Hi, I'm Gerry Fink. My talk is a tribute to Fred Sherman, who I think deserves the title, the First Yeast Molecular Biologist. I'm really sorry that I couldn't join you today in person. Fred Sherman died last year at the age of 81 after a long illness. Fred obtained his Ph.D. with Bob Mortimer at the University of California, Berkeley, followed by a postdoctoral stint with Boris Afouzzi in France and Herschel Roman in Seattle. He spent his entire career as a professor at the University of Rochester School of Medicine. Fred received many awards, including election to the National Academy of Sciences. Now, I was a newly minted Ph.D. the first time that I attended an international yeast meeting here in Seattle in 1965. And it was there that I first met Fred. Now, if you can believe it, the entire meeting had only about 50 people. That was the entire yeast community from all over the world. Seattle, Berkeley, France were then the centers of yeast work. Now, I was used to about the 10 to 12-minute talk, the format you have today at this meeting. So I was a bit unsettled by the organization of talks at this Seattle meeting in 1965. The elders in the field, Herschel Roman, Jack von Borstel, and Piotr Slonimsky, they had no time limits on their talk. Some talked for more than an hour. And what did they talk about? Experiments that didn't work. Dinners they had with other of the reigning elders. And why the next meeting should be in Italy or France, simply because they had fine restaurants that were better than those here in Seattle. One of the people at that meeting, Carl Lindegren, claimed that DNA wasn't the genetic material in yeast. The only person who reported research that we could call molecular biology was a rather bookish, reserved-looking fellow with horn-rimmed glasses named Fred Sherman, shown here in the slide. He seemed rather sober and restrained. But I was to learn that first impressions can be very misleading. Although we now accept the importance of yeast as a model system, the emergence of yeast as a premier organism for molecular biology has often been ascribed to Jim Watson's offhand comment in a 1970 edition of The Molecular Biology of the Gene. There are many reasons to intensify work on organisms such as yeast. Well, why did Watson say that? He saw yeast as the eukaryotic equivalent of E. coli, a microorganism with which one could quickly answer fundamental questions about eukaryotic molecular biology, such as whether the genetic code or the central dogma were universal. To be honest, yeast was not a popular experimental organism at the time, although many of the great scientists had worked on it: Lederberg, Francois Jacob, and Stahl Spiegelman. But their flirtation with the organism was fleeting, and they focused instead on E. coli or bacteriophage, where answers to these questions seem more accessible. It was Fred Sherman, who took up Watson's challenge. He developed the only eukaryotic experimental system that could answer the critical questions of that era about the central dogma, long before DNA sequencing was possible or the term genomics had been coined. These questions were being probed in bacteria by focusing on single genes and inferring the sequence of the corresponding DNA from the sequences of genetically altered proteins. The strategy required a system for facile genetics and a corresponding protein that could be easily sequenced. In eukaryotes, only Fred's ISO1 cytochrome C system had these features. In the late 60s and early 70s, Fred devised an ingenious method for identifying CYC1 mutants and their revertants, along with a remarkably simple, almost one-step purification of the ISO1 cytochrome C protein. Even by today's standards, his achievement is astonishing. I mean, he isolated over 500 CYC1 mutants and determined the amino acid sequences of more than 3,000 variants of the protein. The discoveries revealed by Sherman's CYC1 system were stunning. And perhaps even more important, it brought yeast to the attention of world science. These studies enabled Fred to deduce that the genetic code in eukaryotes is the same as that in bacteria, as were the chain-terminating mutants UAA, UAG, and UGA. This slide shows how he reverted a mutation and used the revertants as a way of deducing the code. So he would start with a mutation like this CYC19 mutant, and then looking at the revertants, all the amino acids, by sequencing the CYC1 protein, he was able to deduce that the original mutation was a UAA codon. And similarly, he did the same process, again, sequencing mutant proteins, and deduced that CYC179 was an amber. Remember, this was all before DNA sequencing. His determination of the DNA sequence of the CYC1 gene, and then actually the CYC1 messenger RNA itself, in collaboration with Jack Szostak and Ray Wu, was a masterpiece. They published this paper in Cell in 1981. And again, remember, this was all done before DNA sequencing. For more than a decade, Fred's CYC1 gene provided the picture of the eukaryotic genome, and Fred's work and findings attracted hordes of new scientists to work on yeast. And, it should be said, Jim Watson to inaugurate the yeast course at Cold Spring Harbor, making yeast the premier eukaryotic model system. Now, I had the opportunity to observe Fred every summer for 17 years. This slide shows the two of us when the course was initiated. He and I taught this Cold Spring Harbor course, a 3-week boot camp of nonstop immersion in yeast molecular biology. Many scientists trace their subsequent success to Fred's dedication to teaching yeast technology. Both of this year's Nobel Prize winners took the course. This slide shows Randy Schekman taking the course. Next to Big Ty, there's Tom Petey's. That's me. And so did Jeremy Thorner, one of this year's awardees, as well as many other senior scientists. Now, as I mentioned, Fred had a very sober, formal demeanor in his scientific presentations. But in contrast, outside the lecture hall, he had a wacky and wild sense of humor. Even a phone call to him turned into a comic routine. If you asked politely, "How are you, Fred?" he would answer, "Oh, I think I'm wonderful, but not everybody agrees with me." Fred had a formal sense of how to give a talk, but a very informal sense in public. During the Cold Spring Harbor course, he would work from early morning until late at night, and that really set the standard for the students. Now, the exception to this dogged dedication to hard work was his conspicuous absence from the Cold Spring Harbor grounds for an hour or two every day. And I was curious about this mysterious disappearance. So one day I asked him, where do you go? He answered, "I take ballet lessons at a studio in Huntington." Fred loved to dance, and he decided to refine his social dancing by learning the arabesque and plie. But his ballet poses did not end in the dance studio. While conversing, he would often move from one balletic position to another, some with an unanticipated, abrupt extension of his leg or arm. Sort of this. These ever-changing balletic poses, coupled with a stream of amusing quips, became his standard form of communication. Although this performance brought smiles to the faces of the students, it made it difficult to concentrate on what he was talking about, as these series of acrobatic moves clouded one's understanding. Now, when my obituary of Fred was published in the journal Genetics, several former students from the course reminded me that Fred's dancing wasn't only formal. In fact, they reminded me that after a long night's work in the lab, Fred would announce, "Let's go dancing." And Fred took the students in the class to a local bar late at night for merriment and social dancing. Here is a reminder that I received from a former student about my obituary. "Very nice, Gerry. I will always remember Fred going up to strangers, always women, usually in bars, and offering them $5 to dance with him. It often worked, but I didn't realize until I read your piece that he was professionally trained." Fred Sherman was an unusual individual. His science paved the way for the blossoming of yeast molecular biology. His humor entertained and delighted many. We owe him a great deal. He deserves the title, the founder of yeast molecular biology.